Hello everyone, my name is Brenda Ramos and today I am presenting an article titled A pH-sensitive motif in an outer membrane protein activates bacterial membrane vesicle production by Rushika et al. This study was published in August 2024 and was a collaborative effort between 7 researchers from different states in the US. The focus of the study is how outer membrane vesicles in gram-negative bacteria are activated. In the image on top is showing a gram-negative bacteria and some of its basic components, such as its cell membrane, peptidoglycan, porins, etc. And then you can see the outer membrane vesicles. So outer membrane vesicles are tiny spherical structures that form from the outer membrane and are released into the environment through a process called outer membrane blebbing. Outer membrane vesicles can play a role in quorum sensing, biofilm formation, immune evasion, and several other processes important for microbe-host interactions. The formation of these vesicles can be triggered by various stressors or factors that affect the structure of the outer membrane and the cell envelope. Some of its constituents are shown on the bottom right image, which includes lipids, proteins, LPS, RNA, DNA, and sometimes endotoxins. Now, in this study, they focus particularly on PAC-C. PAC-C is a 162 residue protein that increases OMV formation as indicated by earlier studies that were key in this one, such as the study by Kitagawa et al. 2010 and Dickenwall et al. 2022. In these studies, they also found that an increase in PAC-C expression results in increased and accelerated OMV formation in Salmonella enterica, Serovar typhimurium. And a model of the topology of PAC-C is in the outer membrane is shown on the bottom left figure. You can see the 4 extracellular loops and their sequences. However, the molecular mechanisms of PAC-C on outer membrane vesicle production were not completely understood, so that's what this study aimed to find. The hypothesis was that PAC-C mediated vesiculation becomes activated at mildly acidic pH, where histidine becomes protonated, thus revealing the mechanism by which PAC-C activates outer membrane vesiculation. So in this study, they used Salmonella enterica, subspecies Enterica Serovar typhimurium, or STM, because it is a facultative intracellular pathogen that is known to increase outer membrane vesicle production inside the acidic vacuoles of host cells. So this makes it a good model to study how pH changes influences outer membrane vesicle production and the underlying mechanisms involved. They also used various different PAC-C RCK chimeras throughout the study to create specific mutants. And these chimeras were detected using rapid anti-PAC-C antibody at a 1:500 dilution, and the expression of mutant protein in the bacterial outer membrane was checked by Western blotting techniques. Substitution of amino acid residues with basic side chains in EL2 and EL3 of PAC-C was done by site-directed mutagenesis, and the sequence of the PAC-C allele mutations were confirmed by DNA sequencing. Lipid A analysis was performed using mass spectrometry and gas chromatography. Cell-based assays of OMV production were performed to assess the impact of pH changes on OMV production by STM, and isolated outer membrane proteins were separated by SDS-PAGE and electrotransferred to a nitrocellulose membrane for Western blotting. One-way ANOVA was used for statistical calculations and molecular dynamic simulations, were performed using Charm 36 force field in order to better understand how PAG-C components interact with its environment. Since outer membrane formation is linked to LPS remodeling, they wanted to examine whether PAG-C chemically modifies STM-LPS. To do this, LPS was purified from wild-type STM and PAG-C deletion mutant bacteria, or OMVs, that were grown in either FOP Q-inducing conditions, which is the 5.8 L media, containing a pH of 5.8 and 10 micromolars of magnesium, or non-FOP Q-inducing conditions, which is the 7.6 H media, containing a pH of 7.6 and 10 millimolars of magnesium. FOPQ, if you recall from our class lectures, is a two-component system that is triggered by changes in magnesium levels in bacterial cells, but also by pH and cationic antimicrobial peptides. And FOPQ promotes upregulation of PAG genes in STM in order to enhance its intracellular survival. Figure A is showing the structure of the most common form of STM-LPS. You can see lipid A in blue, and this is what they mainly focus on in this particular part of the study. If you all remember, lipid A contains fatty acid tails and a polar head made of D-glucosamine residues. And then you can see the rest of the structure of LPS. The green is the inner core and the pink is the outer core made of different sugars. Then you see PAG-L and PAG-P, which are other PAG genes and are also considered outer membrane proteins, shown on the bottom. PAG-L removes an acyl chain from the lipid A moiety of LPS, and PAG-P adds an acyl chain. Now, they wanted to see if there was a change in the LPS structure from wild-type and mutant PAG-C cells after growing them under FOPQ-inducing conditions. But if you look at the mass spectrometry shown in Figure B, both types of cell produce very similar results, indicating that the LPS was similar in both. The lipid A modifications that took place are shown just to the right of that. Figure C is showing the gas chromatography of LPS from, again, wild type versus mutant STM at different pH, so at 7.6 and 7.8. LB stands for lysogeny broth. On the x-axis are the different acyl moieties, which are crucial for understanding lipid A modifications, and on the y-axis is abundance and percentage of OMVs produced. However, again, you can see that there's not much of a difference for each of the acyl moieties between the wild type and the mutant STM-LPS, indicating no differences in lipid A acyl moiety chain modifications were detected in the LPS from both types, further indicating the PAG-C does not affect the acylation profile of lipid A. Lastly, Figure D is showing the SDS page analysis of LPSO antigen from the wild type and mutant STM, which showed similar migration patterns. The bands shown represent different lengths of the O antigen polysaccharide chains, with shorter chains migrating faster and appearing higher on the gel, while longer chains migrate slower and appeared lower. Overall, this demonstrates that the LPS in the wild type and mutant STM is similar, and PAG-C expression does not modify the lipid A moiety of LPS in the outer membrane. Now, they wanted to test whether the extracellular loop sequences specific to PAG-C are responsible for OMV production by comparing its capacity to promote vesiculation to that of RCK's capacity. RCK is an outer membrane protein that belongs to the same protein family as PAG-C and shares a 53% sequence similarity with PAC-C. However, unlike PAC-C, RCK has not been found to enhance OMV production. So here they engineered a series of plasmid-encoded PAC-C RCK chimeric proteins or fusion proteins where the EL2 and EL3 sequence from one protein replaced the EL2 and EL3 sequence of the other protein, and it was inserted into a mutant strain. In figure A, the PAC-C RCK chimeric proteins are shown on the x-axis, and the number of OMVs to the power of 8 produced are shown on the y-axis. PRS1, shown first, is the empty plasmid and serves as the negative control. PPAC-C is the plasmid-encoded PAC-C, which is the wild type, followed by plasmid-encoded RCK, then plasmid-encoded PAC-C RCK23 chimera, in which the EL2 and EL3 sequence of PAC-C was replaced with RCK's EL2 and EL3 sequence. The fifth one is the plasmid-encoded PRCK PPAC-C23, in which the EL2 and EL3 sequence of RCK was replaced with the EL2 and EL3 sequence of PAC-C. Then the rest are the plasmid-encoded protein chimeras, except in the last two, only one sequence of loops of EL2 or EL3 of RCK was replaced with that of PAC-C, EL2, and EL3 sequence. The main things I want to point out from this graph is that the strain expressing the wild type PAC-C produced more OMVs than the strain with the empty plasmid and the one with only RCK, and the protein chimeras in which PAC-C's EL2 and EL3 replaced RCK's EL2 and EL3 sequence also caused an increase in the number of OMVs. So as you can see in the fifth one, and then also in the last bar shown there, except here only one PAC-C EL sequence was used to replace the EL sequence of RCK. However, you can see that it led to a significant increase in the number of OMVs. So these results show that EL2 and EL3 sequences of PAC-C have a significant influence on OMV production, with EL2 in particular having the greatest effect, and that the extracellular loops of PAC-C are responsible for OMV production. Figure B is showing the alpha fold structural model of PAC-C and the outward-facing basic residues or side chains on the loops in yellow, and this was accomplished using structure-based sequence alignment. In figure C, we get another look at the 4 extracellular loops of PAC-C, but also of RCK and that of L. Again, these are all outer membrane proteins that belong in the same family, and here you can see the different side chains in different colors and the chain similarities in all 3 proteins. The blue are basic side chains and the red are acidic side chains, and the yellow represents cysteine residues. And in L, the basic residues were previously shown to establish polar contacts with LPS, which affected the outer membrane structure and enhanced survival on Yersinia pestis. So next I wanted to see if this was the case for PAC-C as well. So to better understand the role of the basic residues in PAC-C, they performed MD simulations to help predict the behavior of them over some time. So here is the initial model of histidine neutral PAC-C in STM outer membrane, figure A. And you can see the different parts of the LPS labeled on the side. So you have the phospholipid lipid layer, lipid A, inner core, and then the outer core. And then in figure C, it is just a closer look. And then in figure D, this is after many nanoseconds later, where histidine is still neutral, just now at a slightly different angle. And then figure E is showing histidine protonated PAC-C with a slightly different conformation, mostly in the EL2 and EL3 loops. In all the models, you can see histidine and phenylalanine side chains represented by the yellow sticks and the basic and acidic side chains represented as just sticks. The bottom graph is showing the time average of RMSF, or root mean square fluctuation of carbon atoms in the loops on the y-axis. RMSF is a measure of the average deviation of atomic positions from their mean positions over time. And that is shown over the PAC-C residue number on the x-axis and the corresponding loops shown on top of figure F. So this data showed that there were multiple interactions between PAC-C and LPS. Specifically, they found that basic residues form hydrogen bonds and electrostatic interactions with lipid A, KDO, the inner core and outer core of LPS, and that acidic residues of PAC-C form polar contacts to LPS phosphate groups. And they did have a supplementary figure showing this. However, I was not able to get it, unfortunately. Together, these interactions help form and stabilize the PAC-C LPS assembly, which could affect membrane remodeling and ultimately OMV production. Therefore, basic residues in the extracellular loops of PAC-C establish polar contacts with LPS and are important for OMV production. However, similar basic residues in RCK and L do not activate OMV production, so they wanted to see why this was the case in PAC-C. So they analyzed them even further, and they found that PAC-C contains 2 amino acid segments with inverse homology and 3 histidine residues unique to PAC-C, which include H60, H62, and H102 in the EL2 and EL3, with a pKa, or acid dissociation constant, of about 6, which makes it susceptible to protonation. Figure A, B, and C are showing, again, the number of OMVs to the power of 8 produced by histidine mutants that were either in plasmid-encoded RCK PAC-C2 chimera or in the plasmid-encoded PAC-C, as seen in figure B and C. In figure A, they created a mutation in which either histidine to alanine, which is uncharged, resulted in significantly lower levels of OMVs in 5.8 L medium. So the first 3 are the ones we saw earlier. Again, PRS1 is the empty plasmid, then we have the plasmid containing the RCK, and then the plasmid with the wild-type PAC-C. And then RCK PAC-C2 chimera, in which the EL2 sequence of PAC-C has replaced the EL2 sequence of RCK. And so compared to the ones with the mutated histidine, the histidine mutants resulted in much lower levels of OMVs. Then in figure B is similar, but now histidine residues were replaced with lysine, which has a positive charge, and this led to increased levels of OMVs, even more than the wild-type PAC-C. The double mutant is shown last and resulted in the greatest number of OMVs. Then in figure C, they created a triple histidine mutant with lysine in both 5.8 L and 7.6 H media. And again, this resulted in a significantly greater OMV production compared to the wild type plasmid-encoded PAG-C in both media. The data from these graphs suggests that PAG-C-mediated vesiculation becomes activated at mildly acidic pH, where histidine becomes protonated. However, since the pKa value of histidine has been shown to vary between 5.6 and 7.7, the researchers believe that wild-type PAG-C may need a lower pH to fully protonate all three histidine residues. And so, for example, in macrophage vacuoles, where pH may range from 4.4 to 5.3, PAG-C in that case may resemble the triple histidine lysine mutant results more closely. Figure D is showing 3 MD simulations of how PAG-C with protonated histidine residues in red and neutral histidine residues in blue affect the histidine-histidine distances across EL2 and EL3. On the x-axis, we see the distance between a particular location of one histidine and another. So in the first graph, between residue H60 and H62, and then between H60 and H102, and then third shown there, H62 and H102. And the probability of it occurring shown on the y-axis. The main thing to note here is that the histidine protonated PAG-C causes a large increase in histidine-histidine distance, which indicates that the conformation of EL2 and EL3 is influenced depending on the environmental conditions. Figure E and F further confirm this. So E is showing EL2 and EL3 in a neutral pH, and here it takes on an overall cylindrical shape. And in figure F, EL2 and EL3 are shown in an acidic pH, where you can see that the electropositive histidine-histidine repulsions decouple EL2 and EL3, and the protein becomes more deeply embedded in the outer membrane, and thus it was concluded that histidine protonation alters the profile of PAG-C interaction with the outer membrane. In summary, OMV formation is generally thought to be driven by the insertion of curvature-inducing molecules, such as modified LPS in the outer membrane, loosening of the outer membrane interactions with the underlying peptidoglycan, and outward turgor pressure resulting from the accumulation of peptidoglycan fragments and misfolded proteins in the periplasmic space. However, in the study, PQ-induced PAG-C is shown to enhance OMV production independently of LPS modifying chemical activities such as PAG-L and PAG-P, and instead may drive membrane curvature and vesicle budding by altering its shape in the outer membrane of gram-negative bacteria. And again, as we learned in Module 4, the PQ system in Salmonella species or other gram-negative bacteria can be activated by changes in extracellular magnesium levels and protons. And then activation of the PQ system then turns on other various systems. However, unlike PAG-L, which leads to LPS modification, PAG-C does not do this. It was also found that EL2 and EL3 of PAG-C are critical for OMV biogenesis, as they contain basic residues that establish multiple interactions with the surrounding LPS. These basic residues result in histidine protonation, which plays a central role in pH sensing by proteins in various physiological settings. For example, viruses that infect via endosomal uptake are known to rely on histidine-based sensors of local pH to activate protein conformational changes that then allow them to fuse with the host membrane and penetrate into the cytosol. Most importantly, these results suggest a model for the way in which PAG-C senses the pH of its environment and promotes OM vesiculation. And understanding how specific outer membrane proteins can direct OMV production is important for the development of vaccines and designer OMVs, as OMVs can be used as a method to deliver antigens, acting as both a vaccine component and an adjuvant. And that's the end of this PowerPoint. Hope you all enjoyed and learned something new. Thank you for watching, and I look forward to answering your questions.